You just joining, can everyone find me and pin me? And thank you for starting the recording. Perfect. So we'll get started. So today we have a presentation of the MC Media. Tim will start with his presentation, and I'll start, and then we'll do a lab tour, and then we'll do a demo of the MCP Media. And by the way, my name is Shikeba Shirzai. I'm an application and training specialist for biomonitoring, food and beverage. And I'll hand it off to Tim now for him to continue his presentation.
Hey everybody, if you were here last week, I did the granulated media demo, and this week we're going to do MC Media Pad. MC Media Pad is a rapid alternative to classical media and requires 1 mil, and it's a very easy product to use, so let's get started. We're going to talk about MC Media Pad for detection of microorganisms, and then Shikeba. Then we're going to do that lab tour like she said, and then we're going to do a demonstration. Let's go. So these are three varieties of MC Media Pad. We have 5. The Staph aureus is not out yet. It's on its way. We also have coliforms, but most people prefer the E. coli and the coliforms, so that's why I'm just showing these 3 pictures. And I just wanted to get an up-close picture so you could see what the packaging looks like and how it's colorful, which makes it easier to determine the differences between the different media pads. So that's what the outside packaging looks like. Next slide. So they are convenient, rapid, and reliable. And we have a rapid aerobic yeast and mold, E. coli, coliform, Staph aureus, and we have some other ones in the works as well. They're easy to store. They are very thin. You can store them in the refrigerator. You can store them once you're done with them. You can reseal it and store it for 30 days. So they're very convenient and easy to use. Next slide. One of the best things about the MC Media Pad is that they are all color coded. And a lot of things in the Millipore Sigma world are going to color coding, like our Oasis, as well as the MC Media Pad. Oh, can we make sure that we're muted? Okay. And one of the best things about the color coding is when you're in the lab, you won't get mixed up. So that's the top part. We also have a grid on all of our MC Media Pads. Essentially, every MC Media Pad looks exactly like this, with different colors and different designations based off of the flavor that it is. Also, it is a pad. It's a non-woven fabric pad instead of a gelatin matrix. You can use the upper part to label. You could use the lower part to label. There is a grid for easier counting. And then also the edges are hydrophobic so that the sample won't slide off once it's part of the pad. And then it's transparent, so the lid, as Shikeba will show, the plastic cover just lifts off. You add your 1 mil of dilution or solution, and then it's going to go and impregnate the pad throughout the entire pad. It uses diffusion and goes throughout the entire pad, and then you will count the next day. Next slide. So very simple. I just put some pictures here. So you open it with one hand. You can take the film off, and then you're gonna do that diagonally, and then you're gonna add 1 mil. One of the things you don't want to do is add it too fast, because you just don't want any sort of splashing. And there are pipettes out there that are pretty strong. You want to kind of find somewhere in the middle. You still want to be fast, but you don't want to add it too fast, because you just don't want any contamination around the lab bench. So next slide. And then you just put it in your incubator. You can stack up to 20 in an incubator. That's a very shiny incubator. And it's going to save you a lot of space in that incubator. And one of the things also, when you are using a pack and you reseal it and put it in the refrigerator, the film is on there pretty good, so that when you reach in, that film isn't going to be flapping around, causing you to throw away a pad that you accidentally touched with your finger. There are other products where that top is kind of loose, So if you reseal that bag, those pads, you can easily contaminate them. Ours, the plastic film is on the pad pretty well, and it's not hard to remove, but it's just easy to not contaminate these pads. And then after you incubate them, you count them, and there's a good picture of our rapid aerobic with roughly 30 colonies on it, and they're nice and bright red. Next slide. So they come in 100 media pads per box, 4 bags of 25, and we store them in the refrigerator, and they have a long shelf life. Most people use so many of these that the shelf life never really comes into play, but they do have a long shelf life if, you know, so that you don't have to worry about stocking. And then let me move my picture here. Okay. And then you can use your open bag within 30 days. So the reason is that they are a dry pad, and there's not a gelatin matrix or not sort of, I don't want to say wet nutrients, but the way that the nutrient mix is, it allows us to have a longer shelf life. Next slide. So subculturing, like most things, if you usually do want to see what kind of bugs we have growing, you can take your needle, your inoculating needle, or some sort of sterile swab, and you can pick colonies and streak them onto other TSA, SDA, whatever other kind of media that you want in order to get isolated colonies for ID. So the dyes don't interfere with the bug like most classical microbiology. This is a classical microbiology in a different format. So the dyes aren't going to affect the growth of the organisms. Next slide. Certification. So as we all know, AOAC kind of rules, ISO rules in the international and certainly in Europe. But over here, AOAC is the most important body. Even though they're not a government body, they are what all the manufacturers need to do is get an AOAC approval for their stuff. So we have the different approvals. We have AOAC, microval, some ISO. And so we have all the approvals from AOAC, PTM, and the OMA is in process for a couple of them, the yeast and mold and the rapid aerobic. And you can see here, this is just a nice representation, again, using the color code, showing our international standards and the approvals that we have and the studies that we've done. So we've already done all the PTM, and some of them are OMA, are in the process of OMA. Next slide. And that just shows, again, the different standards. We did do some microval with them as well. Microval is another Nordval ISO. They're kind of all somewhat the same in that those are all third-party bodies who validate these types of methods. AOAC is not any different. They're just North American based. So what you would do is you send your product to a specific lab, and they test your product according to a specific protocol, and they determine through statistics whether or not it works. They all basically do the same thing. It's just that they have different methods of doing it. So microval will be a little bit different than ISO, maybe a different organism than AOAC. And so they're all are a little bit different in the way that they do things. And even Health Canada now also has some standards. So these agencies are designed to validate, as a third party, are designed to validate our products for use in your lab, and that alleviates the need for each lab individually to validate any sort of method, whether it be classical, rapid, or other kind of methods. And that's what allows customers to feel confident that the product is going to work based off of being validated by a third party. Next slide. And that was it for the usage and the presentation of the actual product itself. Again, we like to keep these kind of light and not heavy on technical details. It's more of a demo. We do have more technical information if you'd like. We have validation studies that we can show, that we can send you. There's stuff on the web. There's a lot of technical data on the web with different matrices. So if you have any questions or have any further need to learn some more about the technical details, let us know and we'll be happy to help. Did we get any questions? To the left there, Shakeba, under the chat where it's the bolded one, it says Francisco has a question there. I think I can see it.
Where do you see that? I don't see it.
It's up towards the upper left. I can see it. What's the
Okay.
Francisco asked, what's the formulation base of each plate? That is what culture medium is used as a basis for manufacturing. You know what, Francisco, if you don't mind, give me one moment and I will show you. Okay, so Shakeba, I'm going to grab this real quick. I'm going to grab the, I'm going to share.
Okay.
Great. Can you see that okay?
Yes.
Great. So there's our rapid aerobic count. It shows all the ingredients there. It's close to standard plate count, Auger. And then there's our yeast and mold with a unique mixture of tetrazoium salt. Of course, we can't tell you everything that's in them. Some of this is proprietary. And this is, again, the more detailed stuff that we can send you as well. And then our coliform with X-Gal. And again, Francisco, we'd be happy to send this to you. This is our customer presentation. It gets more in detail for this particular presentation, but I'd be happy to send it to you. And then we've got our E. coli coliform formulation. So go. I'm going to send that back to you, Shakeba. Yes, I will definitely send it your way, Francisco. At the end, you had to sign up, and so we have your email. We have people's email from this presentation, so we'll send it your way.
Perfect. Thank you, Tim. So
Great.
we'll go and move on to the lab video. It's a video for the biomonitoring food and beverage lab that we have in Burlington, Mass. And I'll start the video. If you cannot hear the audio, let me know right away, please.
We can hear you.
Hi everybody, welcome to Millipore Sigma. I'm Juliana Lescard and I manage the applications and validations team here. And we are in our facility in Burlington, Mass. This is our North American headquarters. And I wanted to show you our dedicated food and bev applications lab. So down here we have this beautiful view, lots of windows, lots of lights. This is Route 95, one of our major roads. And down here we have this really long hallway with all of these labs, and all of these labs are dedicated to customer work. So we do different testing for customers. It's all dedicated to our customers. And come with me and I'll show you our lab. So in this lab we do different things. We do feasibility studies. So if you have a product that you would like to know if it will work with one of our solutions, you can send it to us and it will be tested in this very lab. We also do method optimization. We do on-site evaluations. We do customer training. So if you ever want to come down to Massachusetts and to hang out with us, you're more than welcome to. And here we have just incubators and refrigerators where we keep our samples. And coming a little bit further down here, we have a biosafety cabinet where we do sample prepping. And this is Brian, our lab scientist.
Hi, everyone.
And Brian runs this lab, so if you are sending samples or if you are working with one of us for applications, you most likely will be working with them. So in coming down here, we have the entire setup from the sample enrichment all the way to sample testing. So down here we have the diluent call, and this one will pre-dispense the media from the bottles for you. So you just select the amount of media you would like to add, and it will automatically dispense it for you, so it's handling free. And then from here, we move down to our homogenizer. It will blend your sample for you. And over here, you have different selections of how fine you would like your sample to be. And to start, it is really easy. You just pull on the lever, and it will get going for you. And then over here, we have our automatic sample prep machine. It's called the Pipette Max. And what's nice about this instrument is that it eliminates human error. It's completely robotic, and it minimizes the sample handling step. Of course, you can also have your sample being prepared manually, but this completely eliminates that handling. So you just pretty much load it up and go. And this prepares up to 72 samples. And then coming down here, you have the GDS that also does up to 72 samples. And it will run the samples for you and display the results here. We'll analyze your results and display it on a graph or however you would like it to be reported. So now I'm going to pass it on to Shikeba. She's going to do a quick MC Media Pad demo for you. Thank you.
Hello, everyone. So that was Juliana with the lab tour, and we had Brian in there as well. So that's our food and beverage lab tour. Now we're gonna move on to the live demo of the MC Media Pad. Just give me a quick second while I switch over. Can everyone see that? This area?
Yes.
Yes.
Perfect.
Yes.
Awesome. Thank you. So as Tim said earlier, we had our MC Media Pad. They're color coded. The other cool thing that I want you all to know is that the individual MC Media Pads are color coded. And also what is nice is each of them have the initial of the organism you're testing for on it, and they have the lot number and expiration. So when we work in the lab, we kind of get very busy where we worry about lots and not getting mixed up and things like that. So this is a great way to keep your lots separated while you're doing multiple tests at the same time. So I'll start with the environmental monitoring testing. For the environmental monitoring, all you have to do is you lift up the MC Media transparent lid, and the way how you lift it up, you only need one hand. So you hold it between the index finger and the middle finger, and you lift up the transparent lid, and you tape it down. When you tape it down to a corner, and you leave it to the amount of hours that you want to test the environment for. So you can leave it for an hour, 2 hours, or 3 hours, however much you want to test the environment. And once you're done with the time frame, you take your 1 ml pipetter, you take your sterile fluid, and you go ahead and inoculate it with 1 ml of sterile fluid. So the one thing that Tim did mention earlier, when you go ahead and inoculate it, you kind of want to do it very slowly where it's not going all over. As you can tell, I don't need any additional tools to spread out the solution. It automatically absorbs everything for itself. And once you're done with that, you will go ahead and close it back up nicely. And for any reason, if you notice that there are any bubbles around it, you can easily get rid of them by using your pinky or index finger without touching the pad. Just go around the border of it, and then now you can let it incubate for the organism you're testing for. E. coli is about 24 hours at 35 degrees Celsius. Rapid is 24 to 48 hours, same temperature, 35 degrees Celsius. Yeast and mold is 48 to 72 hours at 25 degrees Celsius. So that was the environmental monitoring. Now I'll go ahead and show you a demo of if you were to do an environmental swab, how you would go ahead and test that. So you would have a regular swab, and whatever environment you're swabbing, let's say for in this instance, it's gonna be the vortexer. So you go around it just a few times, then you take a lateen broth, 2 ml. You go ahead, put your swab in there for a few seconds. And then you'll take this lateen broth and you vortex it for about 30 seconds. So there's 2 ml of broth in there. And after 30 seconds, you will take 1 ml of the broth. Same procedure again. You'll go ahead and open the transparent lid. And you go ahead and inoculate it. Again, same thing, no extra tool is required. It automatically absorbs the solution and you close the lid. And that's for the environmental swab. And again, we let this incubate for the appropriate time you're testing for. Last but not least, we're gonna do the sample testing. If you had a sample, how would you test it? So we're gonna say this is my sample and we're testing for E. coli, EC. Same procedure again. You'll take your pipetter. You'll open your MC media pad. And now it doesn't have to, just so everyone knows, it doesn't have to open up in the corner. You can always use the middle section and open it, whatever is convenient for you. The thing that we wanna say is that you just need one hand. You don't need to hold it out down with the other hand. It's so easy to use. And you take your 1 ml sample and see how nicely it spreads it out? And then you go ahead and close the lid and you cover it. You get rid of the bubbles if you notice any. So once this is incubated, your MC media pad would look like this. As you notice, there's colonies growing on this. Now you can manually count these colonies, but we have a special device. It's called the Scan 1200. It's by a company called InterScience, and it does MC media reading for you, and it also reads your agar plate, and I'll show you how to do that. So once your MC media is it's incubated, then you put that on a stage like this that comes with the Scan 1200, and just give me a second while I switch over to my other camera. Can everyone see that?
Yes. Yes.
Okay, perfect. So as you can see, it's a nice picture of what was incubated. And now you can pick the media you're testing for. In this case, we're testing for E. coli. And the nice thing about this, it has a dilution factor that if you are diluting your media for any reason, you can put that information there. And then all you have to do is press count. And as you can see, it captures the entire thing for you. Just let me. E. So as you can see on the corner, there are some colonies. If you choose to, per your protocol, you can include them or eliminate them. It's up to your protocol. And then once you press count, down here you have a nice information where it tells you how many media was counted, how many colonies were counted, and CFU per mil. And let's see, and what organism you tested for. So it has all that information. You can save this, print it for your data, and present it to your team. Now, very quickly, I'll just show you if you were to read the auger plate on this. So the stage comes off and just the auger plate goes on. And then
Shakeba, let me add that this can be networked into LIM systems. So if you have a LIM system and you want to save this information into your LIM system, there is a networking protocol.
Correct. Thank you for reminding me that, Tim. So let's see. So in this case, if you want your auger plate to be read, you can do the same thing. And then there's new data here. And again, you can expand the colonies however much you want. And then you can go from there. So that was the demo, everyone. Do we have any questions?
Not yet.
Not yet. Okay. Did it make sense to everyone? It's quite easy.
Yeah, it was great. You know, the reader demo was very useful, I think, for people to see, because that's a question. You know, that's something that we can't demo right now. So I think that's a good demonstration.
Absolutely. And it's fairly easy. You just turn it on and you connect the USB to your laptop, and it counts the colonies for you. So of course you can, like I said, you manually can do it, but this instrument, it can count it more accurately where human error is eliminated if you need it.
And not only that, you know, counting 50 plates in a day, it's really tiring on the eyes. So if you are doing a bunch and you do have a lot of